You probably gathered we covered a lot of ground on Friday. And because of that, we're in pretty good shape, so that's very good. I haven't decided where to cut off the material yet. I will announce that probably tomorrow. I may cut it at the end of today, but I haven't decided, so I want to see where things play out. I also haven't scheduled a review session, but I'm planning to do one on Wednesday evening sometime. Location and time to be decided. It will probably be about 7 o'clock, and it will probably be an ALS 4001, but I probably don't know for sure. Okay? So I will hopefully have all that nailed down by tomorrow. I also apologize, I haven't gotten the video and notes posted from Friday yet. I will. The video is ready to go. I should have it up on YouTube shortly, and I'll get today's up as quickly as I can as well, because I know you want to study and get ready for the exam on Friday. We do have an exam in here on Friday. The exam is in here in this room, and pretty straightforward. I've already given you all the answers, so now all you have to do is just match them with the right questions, right? That's a joke. Boy. It's a very tough audience. Okay. So I want to finish up talking about the considerations of enzymes. And one of the things that I'm hoping that you're taking away from what I'm saying about enzymes is that we're very interested in how fast they work. We'll spend some other time later understanding how they're controlled and how it is that they accomplish doing as fast as they do the things that they do. Okay? But what we're talking about here today is really focusing pretty much on how fast they work. Okay? Now, one of the things that I'm going to talk about later in the lecture today has to do with enzyme inhibitors. And enzyme inhibitors turn out to be very important when we think about drugs and stopping enzymes in a bacterium or stopping enzyme in a virus from functioning. And so that's something that I'll talk about after I finish up a couple things up here. Okay. Well, last time I talked a lot about this idea of velocity versus substrate concentration. And I hope that what I impressed you with was that as I increase the substrate concentration for an enzyme, what I do is I make it more likely that the enzyme is going to bump into substrate. At low concentrations of substrate, the likelihood that they bump into each other is low, and consequently, if the substrate doesn't bump into the enzyme, the reaction won't be catalyzed. But if it does bump into the enzyme, the reaction will be catalyzed, and so the more bumping into enzyme occurs, the more likely that the reaction will go. And so that's simply a function of how much substrate we have, that is the concentration of substrate. So we see as the substrate concentration goes up, the velocity goes up. We also saw that the velocity flattened out, and the reason that the velocity flattened out, meaning that the velocity is not increasing very much as we're increasing the substrate concentration way out here, what that means is that the enzyme is getting saturated with substrate. Substrate's bumping into the enzyme all the time. The substrate is not a limitation for the enzyme. And so we've saturated the enzyme with substrate. When we saturate the enzyme with substrate, we reach a velocity known as Vmax, or maximum velocity. And I also introduced the idea at the end of the lecture last time talking about what Km is. Okay? So Km is depicted in this slide. And I'm going to say a little bit more about Km today. I was a little hurried in getting to Km at the end of the lecture last time. So Km is an attempt for us to understand how well the enzyme binds to its substrate. How well does the enzyme bind to its substrate? How much affinity, that's a word we use, how much affinity does the enzyme have for its substrate? A high affinity means that the enzyme binds its substrate very well. A low affinity means that the enzyme binds its substrate very poorly. We get back to relationships. You can imagine a relationship where your significant other is someone that you're very happy to hold on to. Okay? Maybe the significant other has bad BO, and when that happens, the significant other you may not want to hold on to quite as tightly. Right? Or perhaps there are people that you know that I really don't want to go hug them. Right? You know that as a lower affinity. Now, with an enzyme, that affinity is measured by this parameter known as Km. And I told you that Km was the substrate concentration that gave half of Vmax. It was not half of Vmax. It was the substrate concentration that gave half of Vmax. You might say, well, why do we use half of Vmax and why don't we use Vmax? Why isn't it the substrate concentration that gives Vmax? And the answer to that is, you can see that the velocity gets very, 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 very close. We never actually technically reach Vmax. Vmax wouldn't be something that would be very useful for us as a way of measuring how much the enzyme likes its substrate. It turns out that half Vmax is a perfect measure, and I won't go into the thinking here. It's actually mathematically derived. But suffice it to say that half of Vmax, the substrate concentration that gives half of Vmax, is the Km. Now, I want you to think about two different enzymes. All right? Two different enzymes, the same substrate. All right? You got the same substrate. You got enzyme A and you got enzyme B. Okay? Enzyme A binds its substrate very well. It loves to grab a hold of that substrate. No BO or anything like that. Enzyme B looks at that substrate and says, ugh. Right? Doesn't want to bind it. Right? Which one's going to have a higher Km? So let's think about what Km is. Km is a measure of the affinity, and Km is a concentration. If the enzyme does not like its substrate, won't it take more substrate to get it to half Vmax? The answer is yes, it will. You said B, and you're right. All right? So high affinity is the same as low Vmax. It doesn't take very much to get it to half Vmax because the enzyme likes it. Low affinity means a high Km. It takes much more to get it to Vmax over 2. So I'm going to repeat that. I think I may have said it wrong just to start. Okay? High affinity, low Km. Low affinity, high Km. There's an inverse relationship between the Km and the affinity. Yes? High affinity, low Km. Low affinity, high Km. There's an inverse relationship between the affinity of the enzyme for its substrate and the Km. Okay? All right. So that's what Km is. Please don't tell me on an exam that Km is Vmax over 2. You'll make me very happy if nobody does that. But some of you are going to tell me that. Okay. So we understand two very important parameters. The other parameter that I gave you last time was the parameter called KKAT. KKAT was important because I told you that Vmax depends on the amount of enzyme that we use. If I double the amount of enzyme, I will double the Vmax. So I can't compare Vmaxes of enzymes because there's no way of telling how much a person used an enzyme in one case versus the other enzyme in a different case. We need to have something that's independent of the amount of enzyme, and the parameter that's independent of the amount of enzyme is KKAT. I told you that we calculate KKAT by taking the Vmax that we measure and divide it by the enzyme concentration that we use to do that measure. Because remember, when we do those 20 tubes, every one has the same amount of enzyme. Whatever that amount of enzyme was, Vmax divided by that concentration of enzyme gives us KKAT. And KKAT is a number. Here we see catalase has a KKAT of 4 times 10 to the 7th. That's 40 million per second. 40 million molecules of product for each molecule of enzyme for each second. By the way, you see KKAT numbers vary slightly depending upon the way the experiment is done. But we talk about KKAT as essentially being a constant for an enzyme. KKAT is essentially a constant for an enzyme under a given set of conditions. We see widely varying KKATs. Here's catalase that has a KKAT of 40 million per second, and here's lysozyme that has a KKAT of 0.5 per second, meaning it takes 2 seconds to make one molecule of product. Very different kinds of enzymes, right? Okay. You see on the side over here the Km. And I put it on there for a reason so that you can think about something. And this is something that always kind of catches students' interest, and it's kind of cool. In biochemistry, we talk about the idea of a perfect enzyme. And believe it or not, there aren't many things in nature that are perfect. But one of the things that we discover in nature that is perfect, certain enzymes. Certain enzymes are what we call perfect. And I'd like to bring to your mind the concept of what would constitute a perfect enzyme. If we were thinking about an enzyme based on what we've talked about so far, what would make an enzyme perfect? Any thoughts? Say what? Shape plays a role in that, but in terms of the parameters that we've got, velocity, Km, KKAT, and so forth, what would make an enzyme perfect? It's going to be really fast, right? So it's going to have a high KKAT, right? But a high KKAT alone is not the only parameter. Another thing to think about is how much affinity the enzyme has for its substrate. A perfect enzyme will have high affinity for its substrate, and that perfect enzyme will have a very high KKAT. What we can say is that the ratio of KKAT to Km is very, very large. Because we want KKAT to be high, we want Km to be low. It turns out that there are several enzymes, we'll talk about a couple of them in this class later. There are several enzymes that are absolutely perfect. And when we look at a variety of enzymes, we discover that perfect enzymes have a value of KKAT over Km that's pretty much the same. They get to a certain point in that ratio and they don't go any higher. You can't get any more perfect, I guess, right? Perfection is that ratio that's very maximum. And the question that people asked for a long time was, well, why does it reach a maximum? Couldn't it just get faster and faster and faster and have more affinity for its substrate so that Km got smaller and smaller and smaller, so this ratio just gets big, big, big? Why does it level off at a certain place for some enzymes? And the answer, people have come to the conclusion, is that the limiting factor is not the enzyme, it's not the substrate, it's the water. Some enzymes are so efficient that the only thing that limits them is how fast the substrate can diffuse through water to the enzyme. That's the only limit that they have. And since that is a fixed thing, how fast they can diffuse, that's why this ratio hits a maximum. Now I want to think about a couple things with perfect enzymes. One is we know with perfect enzymes that they literally are perfect. They have evolved to the point where if they suffer any mutation, they will be less efficient than what they are. They have evolved literally to perfection. Any mutation makes that enzyme less functional. Okay? One question that you might think about at this point is, well, why aren't all enzymes perfect? Isn't it in the cell's interest to make all of these things go as fast as they possibly can go? The answer is it's not. I'll give you a really good example. How many times do you guys go to Fred Meyer or the grocery store in a week? Right? If you live in the dorm, you don't. But if you live elsewhere and you have to buy groceries, you go do that, right? And you hop in your car, if you have a car, and you drive down to Fred Meyer or to Winco or wherever it is that you go to get your groceries. And you might speed a little bit getting there, be honest. Okay? I see some smiles. There's definitely some speeders in here. But you're probably not going to drive to Winco at 300 miles per hour. Because there might be a little carnage along the way if you did. Right? So absolute speed has problems, right? And absolute speed for a cell has problems as well. What we're going to see is that enzymes, because they can be so efficient, because they can make reactions go so fast, that they can get out of control. We'll talk about enzymes later that will break down carbohydrates. And they're very good and they're very efficient. But if they are going all hell-bent at velocity, they can break down carbohydrate faster than the cell can use it. And that's going to cause some problems. So it's not in the cell's interest to have all of the enzymes perfect. Some enzymes are perfect for reasons we'll talk about later. But just for the same reason, you don't want to have a Maserati going at maximum speed to get to Winco. So too, cells don't want to have all of their enzymes perfect. Because if all of the cars going to Winco were going there in a fast Maserati, I suspect we would have a few more accidents than we do. Okay? Everybody understand? You understand the concept of perfect enzymes. Okay? So that's a very important thing to keep in mind. Okay. Well, let's talk a little bit about things like temperature. Enzymatic reactions vary with temperature. The reason that they vary with temperature, there's two big reasons. One is that the Gibbs Free Energy for a reaction is a function of temperature. We'll talk a little bit about that later. I spare you too much about the Gibbs Free Energy, but we'll talk a little bit about that later. The other is, of course, that enzymes can denature. Temperature is one of the things that can cause an enzyme to denature. Here's an enzyme that has maximum activity at about 40 degrees centigrade. Human body temperature is 37 degrees centigrade. Most of the enzymes that we have within us, not all, but most of the enzymes that we have within us have an optimum at about 37 degrees centigrade. There's a great experiment, a great sort of joke experiment that people described studying fruit flies. Okay? So the scientist gets this graduate student and says, you know, fruit flies behave like an ideal gas. You guys learned what ideal gases were in freshman chemistry. Fruit flies behave like an ideal gas because they're random in their motion. And you start them at a cool temperature, and they pretty much sit on the side of the box, and you heat them up, and they actually will start flying faster. So the graduate student decides to take and test this. And so starts the fruit flies out and notes that at 0 degrees centigrade, which is freezing, okay, the fruit flies are just sitting there. They're not doing anything. warms it up 5 degrees and notes that they're a little bit more active, and up another 5 degrees and they're a little bit more active. And sure enough, he starts plotting where the fruit flies are flying in the box. And the idea of the professor was exactly right, that fruit flies actually do behave like an ideal gas. As the temperature increases, their body temperature warms up, they start moving more rapidly. But then he noted that when he heated them up to about 45 degrees centigrade, that they ceased to behave like an ideal gas. The reason was they all died and fell to the bottom of the box, right? Well, the same thing is happening with this enzyme out here. It's dying and falling to the bottom of the box, all right? So, enzymes have ideal buffer conditions. That includes pH, includes some other things we won't talk about, but it also includes temperature. Okay? I want to say a little bit about ways of explaining how enzymes do and what they do. And I've already told you a little bit about this already. I've told you that enzymes are flexible, and they're different from chemical catalysts. Platinum as a chemical catalyst is just there. It's not flexible in any way. And because it's not flexible, it can't adapt and change electronic environments, which is what enzymes do. And that's why enzymes are much more efficient. There's two models that we have to explain how enzymes do what they do. And I want to explain both of these to you. And I'll explain them to you in a little technical fashion, and then I'll give you a little summary that's much simpler. Okay? Let's do with the technical first. What you see on the screen is a model on the left. That was the model that you probably learned about enzymes in basic biology and maybe in basic chemistry. It's called the lock and key model. And the lock and key model says the following thing. It says that the substrate fits into the enzyme like a key fits into a lock. And there's a perfect match between the substrate and the enzyme. Okay? How many of you have heard that described before? Okay, everybody's heard that described before. Well, I hate to tell you, but that model's wrong. That model is very good at describing the relationship between the shape of the molecule and the shape of the enzyme binding site. And by the way, there's a term that came up in the song the other day that I hadn't given to you, that I should give to you, and that's the active site. Something that you should know. The active site is the place in the enzyme where the substrate binds and the reaction occurs. The active site. Okay? So the lock and key model that's on the left is very good at explaining the relationship between the shape of the substrate and the shape of the active site that binds that substrate. All right? And we've known for a long time that there is a relationship between these two. But it turns out that this model is wrong. Okay? A much more accurate model for how an enzyme works is shown on the right. It's called the Induced fit. And though this figure isn't the best, it's drawn to try to show you that the enzyme has a binding site that sort of looks like this, but you'll notice that its edges are rounded instead of pointed. Okay? It's rounded instead of pointed. And that as the substrate binds to the enzyme, excuse me, the enzyme actually changes its shape. The enzyme becomes pointed. It starts out with these little rounded things here, and it becomes pointed, as you can see here. What you're seeing is something I've already told you. Enzymes are flexible. The enzyme is adapting to the substrate, and the substrate is helping that enzyme to do that. The Induced fit. Now, what this change in the enzyme causes, and we've drawn it in very simple terms here, But what this change in the enzyme causes is the enzyme, when it's in this state bound to the substrate, the enzyme is under stress. It's under stress. What do you do when you're under stress? Eat Oreos? Drink beer? Not sleep? Right? There's an effect when you're under stress, right? There's a consequence of it, right? And that consequence manifests itself in you taking action in some way. Oreos, beer, not getting sleep, etc. The enzyme has consequences as well. The enzyme's electronic environment has changed. The enzyme's electronic environment has changed. The enzyme's shape may have changed. Okay? Both of those are possible. And because there's stress, the enzyme is going to communicate that stress to the substrate it has bound. Just like when you're stressed and you're around your significant other and you get snappy, you're communicating that stress. The enzyme is communicating that stress to the substrate. What happens when the substrate gets stressed? It reacts. Is that what somebody said? Oh, I missed something, I think. When the substrate gets stressed by the stressed enzyme, the substrate reacts. I gave you an example earlier of hexokinase, and I'll remind you of that example. The hexokinase was the enzyme that bound two things. It bound ATP up here, it bound glucose down here, and the binding of those two caused the enzyme to change shape. It's responding to that stress. The change in shape is bringing these guys together. And when you bring these guys together, the phosphate of the ATP moves on to the glucose down here. The substrates are stressed. The substrates react. Make sense? It could just as easily be an enzyme that has bound to one thing. And the enzyme binds to one thing, and now the enzyme goes, oh, wow, and it starts pulling apart. That could be its response to its stress. As it pulls apart, what's going to happen to the molecule that it's bound? It's going to pull the molecule apart. It's how we can split a molecule. There's all kinds of things that can happen. There's just as many different things that can happen as there are enzymatic reactions. But the point is that the flexibility of the enzyme is what makes all of this happen. The enzyme is adapting to the substrate. It's induced fit. And that induced fit, that change in the enzyme, is communicated to the substrate, which responds by reacting. Now, I told you I would give you a technical explanation, and I would give you a simple explanation. Here's the simple explanation. The lock and key model says that the enzyme binds to the substrate, and the substrate is changed. It doesn't say how that happens. It just simply says that the substrate is changed. Okay? You could almost say the enzyme binds to the substrate and magic happens. It is kind of magic, but that doesn't tell us much about mechanism, right? The Koechlin-induced fit model says that not only does the enzyme change the substrate, but the substrate transiently changes the enzyme. That's the stress we're talking about. Yes. Not only, the Koechlin-induced fit says that not only does the enzyme change the substrate, but the substrate transiently changes the enzyme. And notice I said transiently. Because after the substrate has reacted, it gets released, and the enzyme goes back to the place it was in. It's a transient change. Now, I'm very happy with what you guys seem to be absorbing. These are very advanced concepts. Very advanced concepts. A lot of students in biochemistry will have difficulty understanding those. And if you don't understand those, that's perfectly fine too. Come see me. I'm more than happy to work with you and explain them further to you if you're having difficulty understanding them. But I think, I get the sense looking at you, that you are understanding them. Is that an accurate assessment on my part or not? So the heads are clickers. Yeah, okay. Yeah, I don't like clickers. I think clickers are kind of stupid, all right? Yes, there's a here. All right. All right. So, are there questions about what I've told you? Yeah. Transient means short duration, short period of time. Transient is not something that's permanent. Transient is the opposite of permanent. So it happens for a short period of time, and then it goes back to the way that it is. Okay? A person who we describe as a transient is someone who's in town for a short period of time, right? They're passing through. Other questions? Clear as mud? Okay. All right. So the Koechlin-induced fit versus the lock and key model. Okay. Blah, blah. We don't need to worry about that. Okay. I'll show you something kind of cool. We did a plot like this for hemoglobin. Anybody remember the plot we did for hemoglobin and myoglobin? We plotted percent full of oxygen against concentration of oxygen. And what we saw was that myoglobin had a plot that I called hyperbolic that looks kind of like that, right? And hemoglobin looked different. How did hemoglobin look different than myoglobin? What was the word I gave to the hemoglobin plot? Sigmoidal, okay? It had an S shape, all right? And why did hemoglobin have a sigmoidal plot and myoglobin only had a hyperbolic plot? Anybody know? There was a word that we used to describe the behavior of hemoglobin with oxygen. Anybody remember the word cooperativity? What was required for cooperativity? First of all, what was cooperativity? Anybody remember what cooperativity means? Binding of one molecule favors the binding of others. Right? In the case of hemoglobin, that happened because we had multiple subunits of hemoglobin. But with myoglobin, we only had one subunit. We couldn't have cooperativity because once one oxygen bound to myoglobin, there's no other place to bind anymore. But in the case of hemoglobin, binding of one could change a subunit so that the next subunit bound it more readily, the next subunit bound it more readily, and the next subunit bound it more readily. Cooperativity happens because there are multiple subunits to hemoglobin. All right? Now, I've planted that idea in your head. We're now talking about enzymes. And by the way, hemoglobin is not an enzyme. Remember? Hemoglobin is not catalyzing a reaction. It's just carrying oxygens. All hemoglobin's doing. No reaction. But now we're talking about enzymes catalyzing reactions. And we see, first of all, that this guy looks an awful lot like myoglobin in a V versus S plot. What does this look like? This is another enzyme. This is another V versus S plot. And what do we see? We see a sigmoidal plot. What does this tell you about this enzyme? It has a property that's like cooperativity. Okay? In an enzyme, we don't call it cooperativity. We give it another name, and I'm going to give you the name. The name is called allosterism. A-L-L-O-S-T-E-R-I-S-M. An enzyme that exhibits this property is called allosteric. Now, do you suppose, based on the hemoglobin molecule, that the binding of one substrate is going to favor the binding of other substrates? You would be exactly correct. Just like hemoglobin, the binding of one oxygen favored the binding of others and gave rise to a sigmoidal plot, so too will an allosteric enzyme have the binding of one substrate favoring the binding of others. Okay? Now, allosteric enzymes turn out to be really interesting and really important. We're going to talk about that tomorrow when I talk about controlling enzymes. We haven't talked about control yet. We'll talk about that tomorrow. And control is really critical. Enzymes are powerful. Enzymes are fast. And just like we don't want to have that Maserati, everybody going 300 miles an hour to get down to Winco, we want to have a traffic cop to slow things down. That's what controlling enzymes is all about. We'll talk about that tomorrow. And allosterism is one way of controlling an enzyme. The last things I want to talk about today are two things. One, something called a LineweaverBurk plot. Now, the first thing you're going to see about this, oh my God, look at the equations. Are we going to have to memorize those equations? No. Are we going to have to know how to describe how to do a LineweaverBurk plot? Yes. And it's very simple. Editorial. I'm putting her to sleep over here. This is good. Okay. All you have to do is mention Lineweaver-Burk, and then everybody starts going, oh, man. In fact, if you're having trouble falling asleep tonight, I would recommend just say to yourself a few times, Lineweaver-Burk. Okay? Lineweaver-Burk. Lineweaver-Burk. Start counting. You know, people count sheep. Just start counting points on a Lineweaver-Burk plot. Next thing you know, you're gone. All right. Lineweaver-Burk. All right. What's a Lineweaver-Burk plot? Well, a Lineweaver-Burk plot uses exactly the same data that you use for a B versus S plot. And why does it look like this? The reason it looks like this is we take every number associated with that data, and we take the reciprocal. So instead of plotting V versus S, you'll notice we're plotting 1 over V versus 1 over S. Every number has been made the reciprocal of that. The beauty of that is that when we put those points onto a plot, we get a straight line. And straight lines are very easy to deal with. People don't argue about where that line goes, but I might argue about how you draw that curve. So how would you make a Lineweaver-Burk plot? You take the reciprocal of all of the numbers that you have from the V versus S, and you plot them as 1 over V versus 1 over S. That's how you make a Lineweaver-Burk plot. Okay? The reason that a Lineweaver-Burk plot is very powerful is it gives us precise ways of determining what Vmax is and what Km is. Vmax is the value of the place on the y-axis where the line crosses. 1 over Vmax crosses right there. If I take the value of that and I take 1 over it, I will get Vmax, because 1 over Vmax is Vmax. The place where the line crosses the x-axis is minus 1 over Km. If I take the value of this number right here, I take the reciprocal of it and I change the sign, I will have Km. Because minus 1 over Km is Km. These are precise values. I don't have to argue or figure out where they are on a curve, or where does that curve actually intercept. I read these values off very easily. Lineweaver-Burk plots are very, very powerful for us to understand these two very important parameters, Vmax and Km. Don't worry about the slope. if you know where the axes cross, you're in good shape. Questions about that? I told you the first exams where we get most of our calculations, right? We're nearing the end of all that. Okay. All right. The last thing I'll talk about today is something pretty cool. And it has to do with inhibiting enzymes. Inhibiting enzymes is, as I noted at the very top, was something we do with drugs. Many drugs work by inhibiting an enzyme. Aspirin is a good example. Aspirin works by inhibiting an enzyme that makes pain molecules. If the enzyme is inhibited, it can't make pain molecules, and aspirin is therefore described as a pain reliever because the body isn't making those pain molecules. You take an aspirin for a headache, you take an aspirin for muscle ache, or whatever ache that you happen to have, and that's a very good thing. Okay? We're going to talk about two classes of enzyme inhibitors. And I want to emphasize something here that sometimes students don't get. The enzyme inhibitors that we're talking about here do not covalently bind to the enzyme. They do not covalently bind to the enzyme. So when I say that, what does that say to you about the interaction of the molecule and the enzyme? Transient, there's a good word. Comes on, comes off. Remember we talked about that before? Okay? Comes on, comes off. Now, we're going to see some consequences of that. Let's talk first about a competitive inhibitor. Okay? A competitive inhibitor. The word competitive inhibitor tells us that there's some kind of competition involved. The other kind of inhibitor we'll talk about is a non-competitive inhibitor, and that name would tell you that there's no competition involved. Let's start with the competitive inhibitor. It's the easier of the two to understand. Here's an enzyme bound to its substrate on the top. Here is the enzyme bound to a competitive inhibitor. You'll notice that the substrate and the competitive inhibitor both bind to the same site. You'll notice that the competitive inhibitor has a shape that fits into the substrate site very much like the substrate itself does. Competitive inhibitors will always resemble the normal substrate. Competitive inhibitors will always resemble the normal substrate. They compete. They're competing for binding to the active site. Right? A non-competitive inhibitor binds to a different site on the enzyme. It does not compete, it does not resemble the normal substrate. It binds over here. Now, these two types of inhibitors have fundamentally different approaches to doing what they do. Okay? They have fundamentally different approaches to doing what they do. Let's think about the competitive inhibitor first, and then we'll stop for the day. I'm going to talk about non-competitive tomorrow because I think that'll get a little heavy, okay? Let's think about a competitive inhibitor. Let's imagine that we do that V versus S experiment that we talked about before, where I took 20 tubes, I put the same amount of buffer in each one, I put the same amount of enzyme in each one, and now I'm going to add a third thing to each one. I'm going to add the same amount of a competitive inhibitor to each one. Everybody understand? Now I'm going to add varying amounts of substrate, from low amounts all the way up to very high amounts. Everybody with me? The key here is every tube has the same amount of inhibitor. Let's imagine in tube number 1, I've got 5,000 molecules of inhibitor, and I've got 5,000 molecules of substrate. What's the likelihood that the enzyme is going to be bound to an inhibitor versus a substrate? 50-50, right? That's tube number 1. Let's imagine tube number 20, where I have a very high concentration of substrate. I've got 50 million molecules of substrate, and I still have 5,000 molecules of inhibitor. What's the likelihood that the enzyme is going to bind to substrate? Much higher. 99%. In fact, it's going to be really rare that it binds to inhibitor because there's 50 million of these substrates, right? The enzyme is going to be much less inhibited at high substrate concentrations than it is at low substrate concentrations, right? Well, it turns out if you make the substrate concentration high enough, the inhibitor becomes essentially invisible. The enzyme doesn't see it. What does that say about Vmax? What's going to happen if I compare the Vmax of an enzyme in the presence of a competitive inhibitor compared to the Vmax of an enzyme in the absence of a competitive inhibitor? Are they going to be the same value or different values? They're actually going to be the same value. Because at infinite substrate concentrations, which is what we get when we're at Vmax, there essentially is no inhibitor. It's a very important concept. Competitive inhibition does not change Vmax. Okay? Competitive inhibition does not change Vmax. This is a horrible graph. Okay? Blah, blah, blah. Focus on no inhibitor, purple, plus inhibitor, red, and forget about green. Okay? Now, notice that the two lines cross at 1 over Vmax. That means that the inhibitor, Vmax, is the same as the no inhibitor. They have the same Vmax. Do they have the same Km value? They don't. Is the Km value for the inhibitor reaction higher or lower? It turns out it's higher. Higher Km means it has a minus 1 over value closer to 0. You can do the math to convince yourself of that. Okay, that's a lot of stuff. I'm going to refresh that for you tomorrow, and I'm going to talk about non-competitive inhibition tomorrow, and probably I'm going to cut everything off at non-competitive inhibition. Okay? All right. I do have a song for you. It's a song that I can't sing worth a crap, so I actually have one of my recordings of somebody else singing it. I'm going to play for you. And you can sing along. It's called Catalyze. All right, that's good enough. See you guys tomorrow.